So I guess this has been an idea that's been floated quite a bit in Cambridge for the last couple of years. And this is the first year where we decided, let's just try to do this. And Butz has really been amazing at like pushing this forward. But I think we need, I think the main thing is like just we need more conversation at the interface of biology interface. And I think the reason for arranging it in this way, it was like get some idea of the challenges the biologists are facing, or the challenges the physicists are facing, and then having this forum to just discuss and think about how to put it together. We decided that would be a much better way to like have more talking rather than like necessarily just presentation, which often happens. So hopefully this will become more of a yearly thing if we can make it happen. And so Laura's agreed to give the first of these talks. She'll be telling us about mechanosensing, and I don't think it's pretty obvious that there's a clear role for biophysics in mechanosensing. So I'll let Laura take it.
Thank you. Thank you very much. So I'm really excited to be here, and I've been listening to some of the meetings online and attended one or two seminars. So excited to be here on the other side of the podium. And thank you all for coming. So I'll tell you a bit about what my lab are doing here in Cambridge and recently in Glasgow as well. And we're interested in cell migration and how that applies to various normal, healthy, and also diseased tissues. And so the story that is about mechanosensing particularly in pancreatic cancer, which we've been working on for the last several years. Here are just some pictures of pancreatic cancer cells. Nuclei here are in yellow, showing those cells growing in a polyethylene glycol hydrogel, which is soft environment where the cells evolve. Here's the same cells in collagen 1, where you can see invasive fingers and the cells are crawling out and going in their environment. And we think that the mechanical properties, other physical properties like the porosity and the fibrousness of the matrix are very important for how these cells behave. And I'll tell you a story about how we think that stiffness, expression, identity properties of these cells. And the overall goal, obviously, is to look at the benefit of cancer patients. So I should introduce pancreatic cancer. The idea that in the field is that pancreatic cancer multiple stages. Nothing is perfect with the diagram, but gives you a rough in the case of and others, and they go through stages that are called stadia, which are pre-cancer, and then become cancer that can be metastatic over time. This cancer has a very dismal prognosis. It's only got a very positive once the cancer has spread, there isn't a lot that can be done. So you mean the phenotypes here? So these ducts, what happens is they become that the cells in the ducts become gradually more and more abnormal as those mutations accumulate, meaning at first the appearances, they become very elongated, and then they start to disorganize. So they lose normally their cells are all nicely lined up. I have another slide that'll go over that a bit, and they become more disorganized and they form abnormal contacts with each other. And then they finally kind of pile up and they're growing uncontrollably, as well as not very well able to undergo programmed death. This kind of accumulated pile up of cells. I'm not sure if that's what you asked. Yes, overproliferation and also loss of normal cell-cell interactions as well. Some of the data that I'll show you uses a mouse model where we induce this KRAS mutation to be expressed in the pancreas, and we also induce p53 mutation, which is further potentiating the cancer. And this is expressed specifically in the pancreas tissue of these mice. And so that model progresses through these stages as well. And I'll show you some pictures of those on some of the future slides. So pancreatic tumors are known to be particularly stiff tumors. Most tumors or most solid tumors have some element of pressure and stiffness, and they're often discovered by feeling a lump. And that indicates that there's a stiff part of the tissue. This is just kind of CT scans and then a kind of histology picture of a slice through the tumor, showing staining for one of the extracellular matrix molecules here in brown. And that's just showing accumulation of these matrix molecules around the tumor cells. And basically the stiffer the tumors, so I had a conversation with a surgeon in Glasgow, and he basically said, when this tumor is really stiff, he can feel it, and he can probably get better margins on the tumor when he cuts it out. But actually, those are the worst tumors because stiffness often means that the tumor has also spread. And so we think that the stiff matrix that forms around these tumors can probably induce changes in the cells that actually make the cells behave more aggressively. And I'll show you some data about that. Yep. It does, yes, yes. So, yeah, so in the sort of precancer is not very fibrotic, but then over time it gets more and more accumulation of this stiff matrix. Yep. So it's a conversation between the tumor cells and the microenvironment. And the primary cells that secrete the matrix are fibroblasts. So the tumor cells recruit the fibroblasts, and they start out in the pancreas as stellate cells, which are kind of the normal resident fibroblasts in the pancreas. And then the tumor has a conversation chemically with those cells, and they convert into different types of what's called cancer-associated fibroblasts. And then they have this kind of new secretory pathways that they upregulate and start secreting different kinds of matrix and more matrix as well. So yeah, and so this is also just some data, not from my lab, but from the literature showing increases in stiffness can be detected by multiple methods. So this one is ultrasound, where you can do what's called elastography, where you can measure the stiffness of the normal tissue in the tumor. And probably the most relevant is this graph down here showing wild type. This is from a mouse, pre-cancer, and a full-blown cancer. And showing this is the, don't ask me to explain how this elastography totally works, because I can't, but it's basically ultrasound waves used to detect the stiffness of the tissue based on the reflection of those waves. Question? That's okay.
Since we're talking about the mechanics of morphology is somehow important. And at the moment, I don't have any sense of morphology, so the numbers that explain that the tumor is stiff, but then there is margin which perhaps is where stiff,
yes,
and that's where the cancer is spread. So is that the pre-cancer thinking about the cells that actually proliferating on the margin of the tumor, and the stiff tumor, maybe even necrotic cells that are already behind, but they actually tumor effectively spreading the margin into the cell system?
Yeah, it's a good question. So there is a difference between the margin, obviously, and the center. And the center is often necrotic in these tumors. The stiffness is correlated with the deposited collagen around the tumor, and actually acts both as a restrictor of the movement of cells out and as a promoter of movement out, which is kind of a paradox maybe, or something that has slowed it down our understanding and basically progress in this area is that if you remove the collagen from the tumor completely, you'll actually get more spread of the tumor cells because it's like a cage. But if you soften the collagen, then you'll have less invasion. So it's kind of, you need, my sort of feeling about it is that you need a normal stiffness of the tissue in order for the cells to behave normally. And if you have it abnormal, then they start to do things that are wrong. And so in the case of these tumors, they sometimes have fibers that radiate out of the tumor, and those can be seen as potential highways for cells to grab onto and move along. And they also sometimes have fibers that go around and kind of bind the cells together. So it is a mixture, and cancer is chaos. So there's no one kind of right answer for what's happening in a way, which also makes it more frustrating, difficult study. Yeah, so these are just pictures of over here is pictures of different kinds of cancer. So there's breast cancer, esophageal cancer, prostate, lung, pancreatic, and the blue is a stain showing collagen fibrosis. And you can see the pancreatic basically around. So these are the ducts here, and there's some cells in the ducts. You can see some of these might also be blood vessels, but basically in between where you have ducts and vessels, you have this loads and loads of blue collagen. And if you could see, there would also be fibroblasts in there as well. And other cancer types have this fibrosis as well. It's not only pancreatic cancer, but there's especially a lot in pancreatic cancer. So cartoon to explain what we think is happening in terms of the cells and their interactions with each other. So in a normal epithelium, normal tissue, you would have cells that interact with the matrix. So down here, these squiggly lines are just to represent the normal basement membrane around an epithelium. And these cells here are interacting with that through their integrin contacts. So they have transmembrane protein that contacts the matrix and kind of tells them, this is where I'm sitting, this is the basement membrane, and this is where, you know, my home is. And they also have contacts with each other, which in the tissue makes it balanced, and the cells are kind of orderly in the tissue when they sit there. But then when you have things like mutations in KRAS and P53, you start to have this overgrowth where the cells proliferate too much, they fail to die, and they also form abnormal contacts. They start forming sometimes more connections, and they disrupt their cell-to-cell connections as well. And this can lead to a kind of feedback loop of fibrosis and inflammation, where you have breaching of barriers. So as it progresses, they're kind of more and more fibrosis builds up, but also these tumor cells acquire the ability to secrete proteases that break down this matrix, and they can eat holes in it, and they can climb through the holes and escape. And while they're doing that, there are they also make holes in places where they shouldn't be, including the blood vessels, and they secrete signaling molecules that recruit immune cells and fibroblasts that kind of form this abnormal tumor environment, basically the tumor microenvironment, which promotes aggressive behavior and allows the cancer to continue to grow and spread. So that cartoon is taken from this review here. And if you go to the kind of, oh, sorry, go ahead. Mm-hmm.
It's just because that's why we get
it's, yeah, so there's definitely a loss of polarity. So when you have an activation of RAS and you have P53 loss and other oncogenic events, you lose your apical basal polarity. You undergo epithelial mesenchymal transition, so the cells become polarized in a different way. They become like front-back polarity instead of an up-down polarity. And then they start, that helps them to start moving, basically, because normally epithelial cells have a kind of program that tells them not to move. And that's their kind of gene expression, is basically saying, you know, sit here on this membrane, hold on to your neighbors, and don't go too far. But that all breaks down when these mutations happen. Mm. Yes. I think it does, but it's not as well understood how it works as it is with some of the other like adhesion media, because it's harder to model. There are some people who have modeled the pressure by putting cells actually in a pressure devices, and it does change their gene expression and their properties. So it is possible, it's just more difficult. So a bit less is known, I think. So at the molecular level, we know some of the pathways that mediate the mechanosensing from the matrix to, say, the nucleus. And one of them is thought to be this kind of clutch mechanism that cells have. And they call it the molecular clutch, which is basically the, and this is a clutch that has been originally described as a cell migration machinery. That again, these molecules would go through the plasma membrane, connect this filamentous fiber and actually summarize it, pushes the plasma membrane. But it's not a stage that has a great flow, where the action just flows back. And if you look at a cell that's sitting on a kind of relatively unengaged soft matrix, even when the matrix is not that soft, you can actually see this flow happening in the microscope. Engage these molecules here, which connect between and hydrogen, then you have a kind of tight connection here. This now makes a protrusion to the cell and protrudes forward. And meanwhile, it's connected to the matrix, and they send the traction force to have their needs actually kind of unfold when the force is pulling on them. And when that happens, they send out signals. And that can be from opening up a binding site that might attract a kinase, and then you have a kinase signaling cascade, something like that. This tension actually sends feedback that changes the way the cell behaves, in addition to changing the way it moves.
How's that last part?
I guess the retrograde motion itself might not be so important for the gene expression changes. It's more thought to be either physical pulling on the nucleus that opens up the nuclear pores. There's some papers that say that that happens, and then different molecules gain access to the nucleus that wouldn't normally. But also there's phosphorylation cascades that just get triggered by stretching of some of these proteins. Like this yellow one here is vinculin and talin, and those are kind of folded up large proteins that then can get stretched out. And then you have some cryptic binding sites showing that wouldn't normally be showing. And that kind of triggers some signaling, basically. And then this is a bit complicated looking, so don't worry about the details. But basically, one of the major mediators of the signaling from integrin, from matrix integrin to actin, which is now shown here like this, is this protein called the YAP. And there's a partner protein which is similar to it in sequence called TAZ. And one of the things that YAP and TAZ do is they can go into the nucleus and they interact with a transcription factor called TEAD. And that TEAD protein has many genes that it interacts with and changes transcriptional programs that have a big effect on cell growth, proliferation, cell identity, all kinds of signaling inside the cell is controlled by the TEAD proteins. And this is a highly active field, and there are many studies about how this works. And you might know it as the hippo pathway in normal development, controlling the size of organs and tissues. And so it can be influenced not only by stretching, but also by crowding of cells and pressure and other things as well. But it's very much tied to kind of mechanosensing of cells. And as you may know, so this is a meeting about cell fate. So I thought I should mention that YAP and TAS are implicated in cell fate. And this is just a review that I pulled out about the idea that YAP and TAS can control stem cell fate in many different ways, and they have impacts on things like environmental adaptations of cells, including epigenetic remodeling and metabolism. I'm going to talk about metabolism a little bit in my talk. And also they influence fate change of cells like differentiation status and survival of cells, and in response to assaults like chemotherapy, aging, immune system. Question? You mean for downstream of YAP and TAS activation or?
Mechanosensing.
Yes, I think.
There's kind of two layers.
No, you're right. And I think it's complicated, and it's probably they're both important. And I guess the role, I personally think we're still discovering a lot about how this works and about the role of proteins like YAP and TAS. If you look in the literature, you can read hundreds of papers about YAP does this, and then the other paper says it does this, and then it's very, very, we know all these little bits, but we don't know how they fit together, I guess. And that may be where some kind of modeling would be very helpful. And I think it's probably, it's probably just more complicated than what we can figure out by some of the linear experiments that we do in biology, basically. Okay, so a little bit of data. So we wanted to know whether our pancreatic cancer cells that we work on in the lab are mechanosensitive. And so the way we decided to study this was to look at putting the cells on hydrogels where we changed it as little as possible. We tried to only change the stiffness of the gel, not everything else, because cancer is so complicated that you can't do a very controlled experiment. So we took cancer cells from mice and we designed hydrogels that would represent rigid, medium, and soft. And these were made of polyacrylamide that was cross-linked to fibronectin. And we tried to make sure that we had the same amount of fibronectin linked to each of our gels, so that they would have the same number of binding sites for the integrins, but be different stiffness. And when we put our cells on those gels, we found that they did respond. So these are showing cells with actin staining in white and the nucleus in blue. And you can look down here if those are too small, but basically on the glass is the stiffest matrix with fibronectin on. They spread out a lot. They had very big actin fibers and they were highly motile. And then on this stiffness, 38 kilopascals, which represents kind of the stiffness of skin, and the stiffest, they were also very and a little bit less spread out, but still had quite a lot of actin fibers that you could see in the light microscope. And then on softer cells, 7 kilopascals, cells were less motile, but they still had dynamic actin. And then on very soft, they were in these balls that were resembling what I showed on the first slide. But this is still now in a two-dimensional environment. The cells would stick together more to each other than they would to the surface. And these cells, if anything, represented or resembled a kind of epithelial-like state. And these resembled a much more mesenchymal-like state, which is more like a motile spread out cell. This is what they look like when they're in a dish. So you can see these cells are on stiff and these cells are on soft. And this on soft, the cells kind of clump together. They grow more slowly, but they do still proliferate on the soft, and they move less because they're mostly kind of sticking to each other. Where you can see these guys are kind of filling up the dish basically over time. So we found that stiffness promoted migration and proliferation, and wondered whether that was affecting their metabolism as well. And again, I don't need to reintroduce YAP and TAS, we spent a bit of time on that. But we found that in our cells, YAP became more nuclear as the cells were on stiffer environments. So this slide is just showing, you can look at the insets down here, that basically the nucleus is blue, stained with DAPI, and YAP is green with GFP. And you can see that on soft matrix, the YAP was mostly in cytoplasm here on these two, and then becomes more nuclear as you go stiffer. So we could actually see that happening in the cells, which we put in those environments. So we know that this is changing as we would expect. And this is quantification of those results there when we put the cells in a soft or stiff environment. And then what we also looked at was what was happening to the mitochondria of the cells. And the mitochondria are shown here on glass as little thread-like organelles in the cell, which, as you may know, are the organelles that are involved in production of ATP using oxidative phosphorylation. And we found that when the cells were on stiff or glass, the mitochondria were very elongated, indicating for a biologist that these are active mitochondria, whereas when we put the cells on soft, the mitochondria were much more rounded up, indicating they were possibly less active, and they resembled what happens when we use an uncoupling agent to actually uncouple the mitochondria and make them go inactive. They kind of round up when we do that as well. So what does that mean? Yep. Oh, the shadow basically of this. Yes, yes. Yeah. It probably is, and it's probably also that the microtubules of the cell, I think, are supporting the mitochondria, probably are more condensed when the cells are more rounded up. So it's kind of cause and effect, which we don't actually know. We haven't been able to think of a way to separate those, but yeah, no, good point, good point. So to look at, this is the same picture here, just a stills and a bit higher magnification to see the mitochondrial shape. But we also used metabolomics, which is basically taking the cells and lysing them, measuring the amounts of different metabolites in the cells using mass spec. And this was a collaboration with Oliver Maddox in Glasgow. And we basically looked at the ADP to ATP ratio of the cells, which can give you some idea of their metabolic activity. And we found that on soft matrix, the cells had a lot higher ADP to ATP ratio than they did on stiffer matrix, indicating ATP turnover is increasing as the stiffness of the ECM is increasing. And then with Oliver again, we gave the cells radio-labeled glucose, and we asked where the carbons from that glucose were incorporated along the TCA cycle, which is the mitochondrial reaction to make ATP using oxidative phosphorylation. And basically we found that on glass or stiff matrix, you see the higher bars and the colors here. This is indicating incorporation of carbons into each of the, from glucose into each of the components of these metabolites over time, and showing that the TCA cycle basically is going faster in cells that are on glass. And so it's saying that when the cells are in a stiff environment, their metabolism is cranked up. We looked at unbiased or less biased metabolites in the cells as well, to see whether there were any other metabolites that were changing up or down on soft and stiff. And one thing that we found was that creatine and creatinine were changing in a reciprocal way in response to stiffness. This was interesting to us because creatine and creatinine are a part of a kind of energy loop in cells, which are responsible for recycling ATP. And so this is a highly ancient conserved mechanism that cells have for turning ATP into phosphocreatine. And why would a cell want to do that? Because for one phosphocreatine is less reactive. It's the only creatine that can react. And it's also more diffusible than ATP because it's smaller and less charged. And so it's thought to be a good source of energy storage for cells that then can diffuse into areas of high demand and be used to fuel processes that need a burst of energy. And so creatine is a supplement for sport. And it's well known in muscle that when you have high demand on your muscles, you rely on this creatine shuttle to make the best use of ATP. And so we were kind of excited to see that this was changing in our cells, and we wanted to explore that a bit further. And so just to kind of introduce a little bit more this shuttle in cells. So this is a diagram representing here a mitochondrion, and you have creatine kinase both in the mitochondria and out in the cytoplasm of the cell. And in the mitochondria, the function of the creatine kinase is basically to generate phosphocreatine primarily. And so it takes creatine is pumped into the cell or is diffusing into the cell through this passive transporter. And it goes into the mitochondria and gets turned into phosphocreatine. And then phosphocreatine can then be used, say, to the far then be turned into ATP by plasmic creatine kinase, which is creatine kinase B. So you have these multiple different kinds of kinase interact in the mitochondria or the cytoplasm to help the cell generate ATP sites that are high demand. And we found that in our cells that creatine kinase B, shown here on the Western blot, increases in the cytoplasm of cells when the cells are in the presence of stiffer matrix. So this is just a Western blot looking at levels of creatine kinase B in response to plating on these different matrices. And basically, creatine kinase B is increasing on glass over soft. And so we were kind of excited to think, well, maybe these cells were responding to the stiffness by upregulating this shuttle in order to fuel this higher motility that they were doing. And so then the next thing we wanted to do was test what are the consequences of this creatine shuttle for cell migration and cell invasion. And so one of the ways we did this was to use this molecule called cyclocreatine, which can inhibit the creatine shuttle. And that was a way of turning off the shuttle so that we could see what happens to the cells and their migration. This is, again, slightly complicated experiment, but basically what we're doing here, you've got these different pictures. Each of these are showing one invading finger of a cell, basically. And the staining here is actin, and this is stained with LifeAct, which is a dynamic molecule that binds to actin in cells. And then we're going to photoactivate the actin in the cell and ask how long it takes for the actin to turn over. So how long does that photoactivation last before it goes away? And that happens, you see a green spot at all the tips. And then over time, the green fades away as the actin gets replaced by non-photoactivated actin, just turnover of the actin. And basically, we found that in the presence of cyclocreatine, we slowed down the turnover of the actin at the tips of these invading pseudopods. And this one here is a control with a compound called jasplatinolide, which stabilizes the actin in a very dramatic way. So this is jasplatinolide, actin turnover over time, and this is cyclocreatine at two different concentrations, and that's the control there, showing that when you inhibit the phosphocreatine shuttle, you have a slower turnover of actin in the tips of your pseudopods of your cells. We also looked at what happened to the contractile ability of cells. So this is a traction force experiment done in the lab of Manuel Samaran Sanchez in Glasgow. So you put the cells on top of a gel, which is deformable by the cells, and you put beads in the gel that you can then track with the microscope. And when you put your cells on the gel, they can pull and push on the gel, and you can see the beads moving. And if you take the cells off the gel, they go back to their position, and you can measure by how much have the cells kind of deformed the gel. And basically, we looked at under control and cyclocreatine conditions and found that cyclocreatine can inhibit the cells from deforming these gels. So we think that this shuttle is important not only for actin dynamics, but also for contraction, actin and myosin forces that the cells are making. And then we asked, well, does that have any effect on what the cells are doing? And so these are cells invading in a monolayer assay where you take the cells and you make a scratch, which is shown here in red. These are actually 3 different experiments. I should have bars in between there, but you'll see it when they start moving. And basically, over time, these are control cells and cells with cyclocreatine. And you can hopefully see these cells are closing the gap by migrating into the gel and touching their neighbors, whereas these cells are taking longer to close the gap, basically. And so these cells are slower at invading when they don't have their creatine shuttle engaged.
There's more gaps in the
Here, yes, there are more gaps. So they're kind of holding on to each other, but they haven't closed the gap. And it is true that with cyclocreatine, the cells need to become more elongated. We don't actually know why that is. It might be that when cells move, they also need to release at the rear, and that takes energy as well. And so I suspect they're kind of unable to release as well as they normally do, and they kind of just get longer. But we don't actually know how that works. But yes, they do change the way they look as well.
no quantification.
They are, yes. Oh, of the cyclocreatine. So the cyclocreatine will slow down the growth rate. They will definitely grow slower. But they're still dividing. They're not dying, but it will slow them down. And so that will. With the time for cell division. We don't think cell division is contributing hugely to these, but it's a good point. And the way we usually address it is to have cells, and I don't have the experiment here, but we did do it, is to have cells just individually and look at them moving individually and ask if, are they slower? And these are slower. But you're right, there will be some effect also from them dividing. And in these collective experiments, it's very difficult to separate. Some people put in cyclohexamide to stop them dividing, but that can also make cells sick. So we didn't do that. But yeah, it's a good point. And these are cells invading in spheres that we have embedded in collagen. And so again, you can see that these spheres here with cyclocreatine are less invasive, and they don't make these long fingers that go out into the collagen. Hope you can see that. It's not very contrasty. And interestingly, if we label the mitochondria, when the cells are doing that invasion, they're actually tracking the mitochondria out into the tips of their protrusions, as if they need the energy from ATP that the mitochondria are producing in order to fuel the dynamics of actin and myosin that they need in order to invade. And I haven't put this slide in here, but there is an analogy, we can talk about it after if you're interested, that happens in C. elegans during development that's very much like this, with the mitochondria aiding a normal invasion event that happens. And that was discovered in David Sherwood's lab in the US. Just go through that. And we can also ask whether the cells that are out there at the leading edge of these invading fingers are stressed by labeling their a kinase they have called AMP kinase. And AMP kinase is a kind of cell sensor that is detecting whether or not cells are undergoing energetic stress. When a cell is stressed, it turns on this kinase as a way of signaling stress and making a stress response. And basically what we find if we look at the cells and we stain for AMP kinase, which is here in red, is that the cells that are near the front, so if you look over here's control and the cyclocreatine, control and cyclocreatine, that's 24 hours and 48 hours. Basically, near the cells near the front are more stressed, and cyclocreatine enhances the stress. So these cells are struggling to have enough energy to do what they're doing while they're invading, basically. And there are other papers showing that, again, in development, this can happen during developmental migrations, and leader cells are often energetically stressed. And actually, sometimes the leader cells will fall behind and change places with a new cell that has more energy from having followed to then take over and lead the migration, because leading migration is a kind of energetically expensive thing for cells to do. So finally, we asked also whether this has any relevance to cancer, because I remember, if you remember, I started with pancreatic cancer when I started talking about this. And so we looked at some staining, first of all, of some of the molecules that I've been talking about, which include fibronectin, YAP, CKB, and this is normal pancreas up here, and this is pancreatic cancer down here. This is mouse cancer, and in brown in these panels are the molecules of interest, and here is collagen staining in pink. And so basically what this is saying is that when you have a cancer, it has more collagen, as we mentioned, and it's stiffer. It also has more fibronectin and increased expression of YAP and increased expression of CKB. So we think CKB is actually being expressed in these pancreatic tumors, and that as the tumors progress, so this is normal pancreas, pre-cancerous, just at the edge of starting to be cancerous and full-blown cancer, you have this increased expression of CKB in the tumors. And then if we do a mouse model of this cancer, and we take those cancer cells and we transplant them into the mouse, into the spleen, and we look at the ability of those cells to go from the spleen and colonize the liver, we can compare control cells put into a mouse which has been fed cyclocreatine in its drinking water at a concentration that doesn't make the mouse sick, and cells where we've knocked out CKB altogether. And basically, if you look at the livers of these mice over time in this model, you see that there are fewer metastases in the case of knockout of CKB, and in the case of cyclocreatine in the drinking water. We've put little stars there because it's hard to see, but basically we count the number of liver lobes that have these little nodules on after a certain amount of time in the model. So, yep. Sure.
Where is the tissue? Are there tissue the amount that's available? Would you get migration differing in different types?
That's a great question, and I don't know the answer. So there probably are, like, you mean like different parts of the tissue that are more a rich source of creatine. I mean, the liver is a place where creatine can be synthesized, especially, and so the liver is probably very rich in creatine, and that may be one reason why metastasis often happens in the liver. At least energetically, you could think it might be favorable. It's not really known if that's true, but it's a possibility. So yes, there will be differences, but I don't know which tissues are high and low and which. Yeah, it's a cool question.
Yeah, also maybe at the edge.
Oh, like the edge of the tumor. I would love to know. And I did actually, I thought about trying to form a collaboration with these people who do that imaging mass spec, but it's so expensive, like 10,000 pounds per experiment for one, go of a of a tumor. So if it ever becomes possible to do that, I think that would be awesome to be able to look across a tumor or tissue and say, how ATP, how is creatine changing, how are these things, and phosphocreatine as well. So is there another question or we good? Okay. So just to, I think I'll sum up. I had potential of 2 parts, but I think it's better to discuss. So out of this study came kind of hypothesis, basically, that cells are polarizing their metabolism to match the demand that's happening from their cytoskeleton and their activities that they're doing when they're invading. And so this idea of a kind of balance between energy supply and energy demand. And we think of the actin cytoskeleton. So if this is a cell, it's invading into matrix, actin fibers, the transport, which I didn't talk a lot about, but I mentioned metalloproteases are transported in the cell in order to break down the matrix. Also, actin polymerization is happening out there at the tips to fuel the cell to do all these activities. And these all demand ATP. One thing that I don't have a good idea of or a good answer for is how much ATP and how substantial, how significant is this ATP usage in terms of the cell's total energy budget. I would love to know that. And it might be something that could be, better thought about with some models that we don't have. And then the idea that the cell, in order to meet those demands, it does things like recruiting its mitochondria out into its protrusion. So that might help. It also utilizes this creatine phosphate shuttle, which can help the ATP reach the places where it needs to be. And something I didn't mention was also polarization of things like the solute carriers that pump in these molecules that the cells are using has been observed in some systems as well. And so the cell is kind of trying to balance these things. And one of the major signaling kind of complexes that might be contributing to this balance could be the adhesion complexes that cells form between their integrins and the extracellular matrix. And that is partly due to what we know is happening with YEP and TAS, but there might be other signals that are happening as well that we don't yet know about and would very much like to. So I am now going to come out of, oops, wait. I'm gonna, yep.
How much of this does before the test? How much of this for the direct feedback?
No, good question. And we don't know the answer. One way that we have tried to address it is actually to try and just knocking out YAP. And we didn't see a difference when we knocked out YAP, but these cells also expressed TAS, and we haven't got a double knockout. So we can't answer. I think it will probably only be some, because I don't think the gene expression is everything. I think it's going to be also post-translational and activity level of things. So I think it'll be some percentage, but I don't know how much. So I'm assuming I should probably stop now, but I could go a little bit more. It depends. Would you want to have more discussion? Would that be better?
We have half an hour in detail, so
Yep.
We can go into a bit more. We have this 1 hour discussion after the tutor.
Okay. Okay. So if you still have an appetite, I'll do another 5 minutes then, because I was going to talk about. Oh, there we go. Just get rid of that. So I was going to talk about the idea that cells. So this is a slight switch of gear, but it's related, which is the idea that these cancer cells have this great demand for energy. And one of the ways they need it is to change the way they take in their energy as well. So they don't only rewire some of their pathways, but they also change their ability to take up nutrients. And tumors are often places where glucose is in short supply. And it was discovered by Daphne Barsagi's lab not that long ago that macropinocytosis, or this taking of a big gulp of the media by cells, is a major way that cancer cells can take in amino acids and lipids and things in tumors when they're starving for glucose. And so I think this is also related to the cells having this energy demand and energy budget. And one thing that this lab discovered is that these RAS mutations that tumors frequently have, which pancreatic cancer does have as well, like 90% of pancreatic cancer tumors have RAS mutations, promote macropinocytosis. And that RAC1, which is a molecule that we work on for a signaling molecule to the actin cytoskeleton, is an essential downstream mediator of this macropinocytosis. And so what is macropinocytosis? Basically, it's a way for cells to non-specifically take in things from the outside world. And they can take a big gulp, and I'll show you a movie, take these molecules and digest them, and then use them for building blocks for nutrients. They also use them to recycle back to the plasma membrane, things that they take up from the surface. And this is a very conserved process. You can see here a hydra doing macropinocytosis. There's a cup from the stem or the side of the hydra. There's a dictyostelium amoeba with these big cups, and it's happening in tumors. That's just a picture of a tumor, but it's not really showing macropinocytosis. But this video shows a comparison between a moving cell and a macropinocytosing cell. And I hope you can see that in green here is the actin. In this case here, we put red in the medium, which is dextran molecule that's labeled with fluorescence, so that you can see the uptake of substances when the cell goes gulp. And the cell is just taking a big gulp, and you can see these kind of vesicles coming in as the cell takes a gulp. And basically, something that we thought about is that the cell who's crawling is using most of its actin in this front leading edge while it's crawling around. And this cell is using a huge amount of actin to do macropinocytosis. And it's been observed by a few different groups that cells can't very easily always do both of these things at the same time. And this might just be competition for resources. Maybe there's just a finite amount of actin you need to do one or the other. It could be an energy thing. It could also be a signaling thing. It could also be about membrane tension, meaning that these cells, which are very spread out and using all their actin to kind of contact the surface, might not have enough plasma membrane left to do macropinocytosis. And that's one thing that my lab are interested in figuring out, but we don't have an answer to that yet. And then if we think about the molecules that are responsible, we know that the scar wave complex is a large complex of proteins that is responsible for actin assembly at the plasma membrane. So this complex interacts with a molecule called RAC1, which, remember, I said was a signaling downstream of RAS. And this turns this complex into an active state where it releases a tail that binds to another complex called ARB23 and causes a burst of this actin polymerization like you saw in that cell on the previous slide. And we found this molecule SERI, which is an inhibitor of the scar wave complex by virtue of having sequence homology to this subunit of scar wave and binding to RAC1 in a similar way, but failing to trigger this response. So it's kind of competing for scar wave for RAC1. And SERI is conserved in every cell type which has scar wave also has SERI, except for plants. We don't know why plants don't have it. And SERI seems to oppose the activation of RAC1 in cells. This, again, might be a bit too much to go through all this, but it's basically using a fluorescent biosensor that reflects the activity of RAC1 in cells and showing that when we delete SERI in cells, we have more activation of RAC1, and when we overexpress SERI in cells, you have less activation of RAC1. So it's kind of opposing RAC1's activity. And we found that when we delete SERI and we have higher levels of RAC1, we actually have cells that move a bit more. So this is now individual tracts of cells moving. And here you have cells that are control cells that are moving, but not very much, and cells with deletion of seri that are moving a bit more, reflected here as the cell speed. But what we also found that we didn't expect was that seri localizes. So this is seri A, that cells have A and B isoforms. Seri A localizes very transiently and brightly to macropinocytic cups when cells make macropinosomes. And if we delete seri A and B both from our pancreatic cancer cells, we find a decrease in their ability to do macropinocytosis. So seri seems to be important for macropinocytosis to take place. And we've spent some time trying to understand why, and also what the consequences are for the cells. And one of the consequences is that you end up with cells, if you don't have seri, you end up with cells that have a lot of integrin on their surface. And that's because some proportion of the integrin on the surface of cells turns over by being macropinocytosed. So you have seri A labeling in cyan, and seri B is down here, and then integrin alpha 5 here. And you can basically see, I hope, that when there's a macropinocytic event, you get that bright flash of seri A, and you can see the integrin co-localizing on those vesicles. So cells that don't have seri have more integrin on their surface. I haven't got the data on here because it's too much to show all of it. And they become more migratory, more invasive, and they're able to grow more easily without anchorage. They also have enlarged focal adhesions. So these are control cells and these are seri knockout cells. And that's just measuring the size of the average integrin cluster in the cells. And despite moving a bit faster, they are terrible at chemotaxis. So they don't know where they're going. And this again is individual cell tracks here shown in different colors of seri knockouts, which are basically moving randomly, rescue cells, which then move directionally. This is a gradient of fetal bovine serum. So they're moving towards this direction here. And this is an inhibitor of a receptor, which we know is important for moving towards fetal bovine serum, which is the LPA receptor. So lysophosphatidic acid, which is the kind of main component in the serum that attracts these cells. And this is, in our mind, a kind of in vitro analogy for chemotaxing cells from a tumor to a blood vessel. And this is not perfect, but we know that blood has LPA in it, and that's attractive to cancer cells. And so we think that cancer cells are homing to those places, partly by tracking towards LPA, and that somehow seri is important for cells to be able to do that. And we think that's because the LPA receptor itself is also tracking the same way integrins do, together with these macropinocytic events. And so basically here you have LPAR1, which is the LPA receptor, and then you have dextran, which is labeled to show the macropinocytic events. And I hope you can see they co-localize. And then also you have co-localization of seri B with LPAR1 when you have macropinocytosis happening. So we can see them at the same time in the same place undergoing macropinocytosis. So while we had thought that macropinocytosis was primarily important for this taking up of nutrients, we now know that it's also important for trafficking of surface receptors on the cells that we hadn't really thought about before. I'm going to skip this data here because it's just showing the same thing another way, and say that our next thing that we're trying to do is then look at whether seri is important in cancer progression, because not only have we found it's important for macropinocytosis, but we've also found that it's important for this receptor trafficking, and we know that that's probably important for cancer as well. And that's just for the future. We're planning to use the same model that we've used for pancreatic cancer, but now delete seri in the pancreas and look at escape from the primary tumor and metastasis to the liver. So questions that we have, going back to the beginning of the talk, we want to know how the cytoskeleton, which demands energy, interfaces with the creatine phosphate shuttle. We want to know how Siri proteins might act as buffers of the RAC1 protein and how that affects tumor formation and tumor progression. And we want to know how receptor trafficking might be controlled by macropinocytosis, which is kind of a new spin on what we thought was just a way of cells to take up more nutrients. And we're asking questions like whether these Siri deficient tumor spheres and tumors have a real defect in macropinocytosis. And I need to thank my group for their wonderful work. This is the Glasgow group here, and this work mostly was done by Emily and Savas and Heather. And then I have my new group here in Cambridge, who I now need to get a picture of so I can show them off as well. But thank you very much for your attention.